This will just be an introduction to PyMOL, which is a molecular visualization software that we can use to play around with the structures of proteins and other macromolecules in 3D and look at things like where bonds might form, where important amino acid residues are sticking out, and just get a general sense of a protein's makeup and what regions might be important for doing various things. Protein I'm going to be using as an example is going to be this phenylalanine hydroxylase, which is necessary for breaking down the amino acid phenylalanine. People who have specific mutations in this protein get a disease called PKU, which is phenylketonuria, which makes it so that they can't break down phenylalanine. And so if they eat phenylalanine, byproducts build up and it's bad. And so that's why you have the warnings on foods that say, like, caution, phenylketonuria product contains phenylalanine. And so this protein is actually this tetramer. And it's a homotetramer, so all these chains are the same. Some of the structures that you find only have a single one of these chains. And so you'll see like these just have a single chain, where some of them have all the chains. And some of these only have a portion of the protein. And some of these say they have the whole chain, but really they don't have the full chain that's actually going to be visible. So I'll show you one of these examples. The example that I'm going to use is the 6-HPO for starters, and then we'll go and we'll pull up a tetramer. So I'm going to come over here and I'm going to go to fetch 6-HPO. So that's how you get things in the PDB, is type fetch 6-HPO. And that's going to pull up the structure. Alternatively, I could have come to this top toolbar and gone fetch, get PDB, and I can put in the PDB ID here. It's still using the 4-letter PDB IDs, but the PDB IDs are switching over to this 12-letter code because they're running out of the 4-letter ones. And so that's what this longer thing is. But at least in this PyMOL 3, it's still using the 4-character PDB ID.
PyMOL also allows you to open PDB files or MCIF files, which is a different file format. And so you just go to File Open. And this is really good if you have a custom PDB file, such as if you did some modeling of a protein or something like this, and if you're using an AlphaFold predicted protein. Because PyMOL can't pull in AlphaFold models, but you can come over here. So when you're in Uniprot, if you come over here and choose the AlphaFold, press this, it'll download the PDB file for you. Alternatively, what you can do is you can actually go to the AlphaFold database. And on that AlphaFold database, it'll allow you to download the files. So you actually have a couple different options when you're downloading files for structures. So PDB files are actually kind of the more outdated one. And so they're being phased out by these MMCIFs. So the MMCIF is better, but PyMOL will take either of those file types. Either one of those, download them, and then you go over here and go to File and Open. Or if it's something that is in the PDB and not an AlphaFold predicted model, then you can just use that fetch space and put what you want. Or you can go to File, get PDB, and do that. It also then allows you to choose an assembly or just a specific chain. And so if you want to know more about that, talk to me, but those are options.
Okay, so over here now we have the structure pulled up. We have our basic cartoon or ribbon diagram. We can change the way that we represent it, and I'll show you how we can change the way we represent it. We also see all these weird plus signs. These weird plus signs are just water molecules, and I'll show you how we can remove those. Orienting yourself to this window. So up here we have that basic toolbar. And so here you're going to find your basic things. Like you would see a file, but here some of the file names things are different. One of these is save sessions. So the first thing you want to do in like any PyMOL session is to save it. And so you're going to want to have a folder. And so I'll have my like demo stuff folder. And so you'll have your Chem 370 folder or whatever, and you're just going to go and type in whatever it is. So 6-HPO demo. Okay. And so now I have that. And I'm just going to Command S or save however you want to save it. So if you just want to go save session, you're going to do that frequently throughout. And that's going to help it so that if you make a mistake, it'll only let you undo certain things. And if it crashes or something, which can happen because it's computationally intensive at times, you want to make sure that you've got it saved. And you want to make sure you have it saved to somewhere you know. So save your session and save it frequently. So I have my session saved. Now I'm going to start doing stuff. But I realized I've kind of been moving things around and haven't really showed you how to move things around. It's going to depend on what kind of computer you have and whether you're using a mouse or a trackpad. And so you can go and you can see some of the various commands. I'm using a Mac and I'm using a trackpad. And I'm going to tell you some of the things that I do. And so if I do 2 fingers and then I rotate my top finger, I move around my top finger, that's how I rotate things. If I press Option and then I have 2 fingers, I can just drag and move things, translate it. So I'm not turning anything. I'm just moving it from side to side or up and down. If I do 2 fingers and I scroll up or down, what I'm doing is I'm kind of like slabbing it. And you can also go, if you go to Display, you can go to Clip, and that's going to allow you to kind of define the size of the slab, which is going to allow you to kind of hide some of the details, make a sort of slice through it. So various things you could do, and I'm just going to show you the basics in this one, so I'm not going to go into that. But that's kind of the basics of how you would orient around here. So 2-finger click and rotate your top finger in order to kind of turn things around with your left mouse. If you want to go from side to side, so Option, you can move things around. And then up or down. Oh, to zoom in, you can just pinch and drag or use like your middle scroll bar. So pinch, drag, pinch, drag, pinch, drag on a trackpad. And then 2 fingers up or down to kind of do that slab. And one of the first things I want to do is I'm just going to come down here and come over here. This is going to be allowing me to do various things. One of the things that I always like to do is press this seek button. This is going to show me the sequence. So up here, I can see the sequence. And if I click on something in the sequence, it'll show me where that is in the structure. Right now, I have it set to select residues. This is really important. So if you come up over here on this top left, right now I'm selecting residues. I could also select different things. So if I want to select a chain, now if I click on something, it's going to select the entire chain rather than just selecting a single residue. And if I click somewhere else, so if I click outside of here, it's gonna, then the selection is gonna come off, but it's actually still selected. So if you come over here, this is your object toolbar. And so each thing that you have, like each structure or each object that you define, such as a selection, is going to show up over here. And then you can play around with them separately. And so right now I'm selecting chains, and either I can select it based on the sequence, or I select it down here. Sometimes that can be helpful, but often what you're going to want to do is you're going to want to selecting residues. And so that's what I'm going to change it to. Now, if I click something, it's going to select a single residue. And you can see that the selection is going to show up here. What's a little confusing is that when you have this numbering, it's right now, this is 118. So this is this Bailey 118. When you have the numbers up here, the number starts at like the left. So 116 would be this D, this aspartate. If you're not familiar enough with the 1-letter codes yet, you can also go over here. You can go to Display, Sequence Mode, and then you can actually go to Residue Names, and that'll give you the 3-letter codes. But now you can see it takes up a lot more space, and so that can be harder too. So that's one of the reasons why it's really good to be able to know just the 1-letter abbreviations. One thing you'll see here, although this shows that it has like the entire chain, so it starts at 1 and it goes to 4, whatever it is, you can actually see that there's a lot of it that's grayed out, and it's not even going to allow you to select this region. This region is present in the actual protein that's physically there, but the scientists couldn't get strong enough data to actually be able to see it. And so you can only see it starting at this 117. So even if you click on this grayed out region, you're not going to be able to see it. And so if you wanted to be able to look at something in that region, you'd want to choose a different structure. And I'll show you in a minute how we can go and we can look at a different structure where there might be gray regions in between. So up here, we only have a single chain of this protein in this structure. But you can also see that we have this iron atom. And so when you see one of these spheres, that's often going to be an ion. So that's going to be an ion atom. And if we go and we look over here, these are going to be water molecules. And note that I was moving around. That's just because I had it set to rock. I accidentally clicked rock. So typically you're not going to have rock set. I like to get rid of these water molecules. The main way we're going to do anything is often going to be over here with this toolbar, this object panel.
So we have A, S, H, L, and C, and you're going to see these options for all the different objects that show up, all the different objects and selections. And so A is action, S is show, H is hide, L is label, and C is color. And so we'll go through and look at the different things that we could do with those. And you're going to press the one that is next to the thing that you want to do. So like if I do all of these at once, I want to do the whole like object or just a selection. So we'll get through examples.
A is action. This is where you're going to do various things like you can align, you can find contacts, so look for protected bonds, things like this. S, this is going to be where you can control the representation style. So right now I have it ribbon or cartoon. I can also have it do things like sticks. One thing that you'll see when you go and you do sticks on top of a ribbon, so you see they overlaid, and sometimes it can get a little awkward when you have, say, a side chain that's sticking off of a cartoon. What you want to do is come over here, go to Setting, Cartoon, and click on the side chain helper. That's going to clean things up. So one of the first things that you're going to want to do when you open a PyMOL session is going to go to Setting, Cartoon, and Sidechain Helper. You can also use it during the command bar by typing in set cartoon underscore side underscore chain underscore helper, comma on, or 1. So there are various ways to do things, and typically you can always do things through the command bar, and more things that you could do with the GUI, but you can do it in the GUI as well. So now I have that on, and I still have these water molecules that I'm going to want to get rid of, and I'll do that in a minute. It can be a bit overwhelming to see all of the sticks at once. And what I might want to do is instead go, for now, I'm just going to go and I'm going to hide the sticks. And later we're going to see how we can go and we can actually display the sticks just for specific amino acid residues. It can also be distracting to have all these waters, so let's go back and do some more things. So this H, this is going to be hide. And so I was using hide to go and unshow the side chain residues, but I can also come over here and I can do hide waters. And that'll just make it so that these red dots will go away. So I'm not looking at the waters, but they'll still be here physically in the sequence. If I want to actually remove them, I can go to Action, Remove Waters. It's going to physically delete them. If I wanted to label things, maybe what I want to do is I want to label the chains. And so now you can see that it really only had this one chain, but it's going to now try to label this iron. And now I'm going to go to label and I'm just going to clear that. I can also color things. And so maybe I want to color things by elements. I can also color things by chains, which doesn't make that much sense when we just have a single chain, but I'll show you in a minute how this can be helpful when we have multiple chains. I can do things by secondary structures. So if I want to highlight the secondary structures like our alpha helices and our beta strands, I can do that. I could also do it color based on this spectrum. And then it's going to go from the N-terminus to the C-terminus. So N-terminus would be blue, and then C-terminus would be red. And that'll allow you to see kind of the order in which things go. And note that when I color things, the coloring scheme is represented up here in the sequence as well. One of the final things I want to show you color-wise is you can actually go, and if you go to spectrum B factors, that's going to kind of show you the confidence of the structure, how fuzzy it was. And so you can see that at these ends, where it's getting to that region they can't even see, you're going to have a higher B factor, more kind of movement in that structure. So this was a single chain, and I'm just going to go back and I'm going to go and I'm just going to color it by chain. And what I'm going to actually do is first I'm going to color it by chain. Now I'm going to go and I'm going to color it by element. And it's not going to make much sense right now, but what it's going to do is it's going to color things by element when I have the stick showing. But right now I haven't told it to kind of highlight any features. So I wanted to find my own selection. So when we click on something, so right now I'm in that residues mode. So when I click on something, I'm going to select a residue. I do not want to have it in that chain mode right now because then I'd be selecting the entire chain, and I want to select a single residue. When I select a residue, a couple things happen. One is I see this green thing show up down here. This is indicating the location at which that is in this overall sequence. And you can see that right here. I selected the 148. So I've selected this glycine 148. And so this would be 146, 147, 148. What I actually want to do though is, and then if I click elsewhere, you can see it's actually still selected. But if I go in and make a new selection, now it's going to get rid of that old selection. And I'll show you how we can go and find a specific place we want to select. So going back to some of the things in the PyMOL window, up here, what you'll see is the scenes, and you'll also see this draw array. We're going to be doing some things where we're going to be making some pretty figures. We're going to be getting things colored in an array we really want, and we're going to want to save that. So not only do you want to save our session, which I should do, by the way, so Command S, and I'm going to save the session. But over here, I have the scenes, and so I really like this view, this representation, this coloring. What I can do is I can press this plus button, and it'll save a scene. I could also rename the scene if I want to do various things. Say I want to color a different color. So maybe now I'm going to go back and I'm going to color it by its secondary structure. And maybe I want to actually go and I want to show it as surface. And so I want to show the surface of the protein and not just the ribbon diagram. And it's not very useful to have it by secondary structure right now. So maybe I decide, and instead I want to go and I want to color it back to that rainbow. Okay, so maybe that's what I like, and maybe it's not, but whatever. I'm just going to save a scene for now, and I want to show you that if I click on this, I'll go back to that original scene. If I click on this, I'll come back to this. And so this is a great way to both save things you might want to save as a picture later, as well as to save various representations and orientations. And then if I decide that I want to go and take a drawing of it, I have this draw or this fast option. If I do the draw, it'll just take a quick and dirty picture export for us. If we do this ray, then it'll actually go and ray trace it and make a really pretty looking picture, and you can do a transparent background. Speaking of background, if I go to display, I can go to background and I can actually select set the background to a different background. So maybe I want to do it white, and I have a white background. Now, if I wanted to update the scene, I can just do update like that. And now if I go to somewhere else, I can have this updated. But note that the background is going to kind of be the same for all of these. So it's not going to change the background when you go back to a scene. So that wasn't a great example. But if I wanted to say color it differently, so maybe I want to go and I want to color it blue. And now I want to save it. I can update my scene. Or I could just save a new scene and then I could get back to where it was. Okay, so now let's go and actually do something a little more fun where we select a specific residue and we'll show it as stick. What I want to have us do is, I don't really like this one, so I'm just going to go and I'm going to delete that. I'm going to go and I want to find that disease-causing residue. And so if I go back to my Uniprot, which is where I could find these structures, I can go to the variant viewer and see disease-associated mutations. So a kind of classic PKU mutation is going to be this 408 R to W. So that's going to be a substitution mutation. And it's a missense mutation. So you're changing the amino acid arginine to a tryptophan. If you think about arginine, we've got our basic positively charged amino acid. Tryptophan is going to be one of those nonpolar aromatic. So big change. And then let's see where this change is happening in the protein. So we want to locate position 408. Now, things can be a little complicated because when you go to the numbering might not be what you expect. And so what I like to do is if you go to the sequence, we can see that 408. So this should be this arginine right here. We want to make sure that this numbering scheme is in line with what we see up here. Sometimes what happens is the numbering is a little different in the Uniprot sequence and in the PDB sequence. So if you go to the sequence in the PDB entry, which should match what you see in PyMOL, you want to make sure that the sequence is actually the same. And so if we go to that 408 location, we should hopefully see that the sequence is going to be the same. Okay, so we have this 408 location. Okay, we've got it. The numbering's the same. If the numbering wasn't the same, then you have to kind of take the sequence, find the sequence surrounding the region that you want in the Uniprot sequence, and its accompanying numbering in the PyMOL sequence. In this case, they're the same, so I'm not going to worry about that. Instead, I'm just going to come over here. I'm going to find that 408R. I want to make sure that I'm in viewing, and so sometimes if you were going and looking at the mouse settings, you might have accidentally clicked editing, and I accidentally did this. You want to make sure you're on viewing. And now what I want to do is I want to go and make sure I'm on select residues. I'm going to come scroll over here until I find 408, and I'm going to click on it. So now you can see that it showed up as this celly. This is my selection. When you make a new selection, it's going to be temporarily stored in this celly. What I want to do is I want to actually store it as a separate entry over here in this object panel. There are a couple of different ways I could do this. One is I could use the command bar, and I could go to set underscore name. And then I have my old name. So my old name would be celly, comma, the new name. And maybe I want to do with mute site 408 or something. And now you can see that it changed over here. Alternatively, I could go to action. I could go to rename selection. It's going to show up over here. And now I'm going to delete it. And I'm just going to do 408R site or something. And whatever I name it is fine. But now it's going to show up over here. And if I click away and I click back on it, it should now have it saved. And I can make multiple selections, and each of them would show up as an object over here. Now I can manipulate it separately than manipulating the whole thing. So if I come if I have multiple chains or anything, it's going to change everything at once. If I do it with just the object, it'll change that object as a whole. And if I make a change in just the selection that I have as an object, it'll make a change to that part of a bigger object, but only to this part. So maybe I want to take the selection and I want to color it and I want to show it sidechain. So right now I can see where the sidechain would be because it's kind of highlighted, but it's not actually showing. So if I come over here, go to S for show, and then what I want to do, show as, and I want to go to show as sticks. And you can see that I'm showing it as sticks. Unfortunately, I also then hid the cartoon. And so what I want to actually do is go to show as cartoon and make sure that I'm showing as cartoon and stick. Alternatively, what I could have done is I could have gone to show as side, and I could do side chain and then show side chain as sticks. So if I want to redo, so I'm just going to show you that again. And so if this were, I'm just going to hide this so we can hide the sticks, and we'll just do this again. So if I come over here and I go to show, and then I go to side chain as sticks, now I can show the side chain as sticks with keeping the cartoons. If I just go to show as sticks and I forgot to do it just as the side chain, then I would have to go and back and also show it as cartoon. So you can show cartoon and stick at the same time. And right now it's only showing stick for the specific amino acid residue that I want to highlight. To make it even more obvious, what I'm going to do is I'm going to come over here, I'm going to color it red. And now I have something I like. I want to do a couple different things. One is I'm going to go and I'm going to press the plus and make a scene. And I can come over here and I can rename my scene. Maybe I have mute site stick or something. And now what I want to do too is I want to save my PyMOL session. So I'm going to do my command S. I want to make the iron a different color. So maybe I can do this a couple ways. One, I could come over here and I can select it over here. I can also just click it over here. What I want to do is I want to color this. Maybe I want to color it by elements. And now I like this. And so I want to update my scene. And so I could do that over here. And I want to save my session. And now I want to come over here and maybe I want to make a figure. And maybe I want to make a figure where I zoom in. So I pinch scroll and I come over here and I find an orientation I like. And now I want to do is I want to save a new scene. One thing you might want to do too is maybe I want to label this. So I come over here to that selection I like and I want to go and I want to L for label. And I want to label the residues. And now it's going to show me that residue labeled. And if you go, there's various ways you can actually move the label. So if you go to editing, you go to commands, you can move the label. But be careful with editing so you don't do something wrong. You can also edit, like add the label in something like Illustrator or some other like PowerPoint or whatever you want. And that's how you would make a figure to show a certain mutation site. And note that it can be kind of hard to see where things are depending on your orientation. And so you need to, it's your job as a scientist to kind of find a representation style, find an orientation that makes it so that readers can see what you want them to see. And so this might mean using some of that slabbing. It might mean finding an optimal balance between stick and ribbon and cartoon, and what you show and what you don't show and what you want to emphasize. Speaking of emphasizing things, right now I have this whole side change colored one color. If I wanted to, I could come over here and I could go color by element. And now I'm just going to color the elements. And so you can see that now it has the nitrogen blue and it has oxygen red. It's a little confusing because the residue itself is red. And so that's not the best example, but you could do things like that. And then maybe you want to recolor the rest of the chain so that you don't have that problem. So you can clearly distinguish things. So you come over here and you go to color. You can choose a color. So maybe I want this aquamarine. It's going to color everything this aquamarine. And so first of all, I just want to get rid of the waters. Actually, I'm just going to remove waters. Okay. So it colored everything this blue. Now what I want to do is come over here and color by element. If I click this top one, what it's going to do is it's going to keep that backbone color as blue. So any carbons are going to show up as blue, but then you're going to use this conventional coloring scheme where you have hydrogen's gray, nitrogen's blue, oxygen's red, and sulfur's S. And so then your ions are going to be different colors too. That's one way you can get it ions in different colors. But the nice thing about this too is that then if I go to show sticks, you can see that it kept the carbon backbone in the blue, and then it has the different colors for the different elements. And again, you're seeing this weird stuff because I'm displaying sticks on cartoons because I opened a new session because I forgot to show you this. But if I go to settings and I can go to cartoon, sidechain helper, it'll clear that up. If I come over here, I can change the color and I can change this backbone color. Or what I can do too is if I go to by element, I can choose one of these set colors for the backbone. And then it'll keep the colors of the elements. If you go to setting, some people like shadows, I don't like shadows. And so what I like to do is I will go to rendering, shadows, and I want to do none. And that way, when it does that rendering, it's not going to have shadows. But some people like shadows, so you can have shadows, don't have shadows, whatever you want. That's just how you would do it. Okay, now let's go and let's pull up a tetramer so we have some multiple chains. And so going back to that entry for PAH in Uniprot, if we go to the structure, so right now it's showing us all of the structures that are available, all the PDB files that are available. We can see how they were solved. We can see their PDB ID, their resolution, how much they cover, and how many chains they have. And so if it's just saying A, it's just a single chain. The one we're looking at right now only has the single chain. At the end, you're typically always going to see an AlphaFold predicted structure, and that's going to have the entire thing. But there are regions that are less well confidently predicted, and so those are going to be the orange, and then the regions with more high confidence are going to be blue. And we can see that we are kind of only seeing a portion of our structure before. We are missing this entire front end, and we are only seeing a single subunit. So the AlphaFold is typically only going to have a single subunit too. This structure looks like a good bet for us. So this one is actually a tetramer. It's a crystal structure. It's got all the chains, and it's got almost all full length of the chains. But as we'll see, there's still going to be regions that are missing. Let's go ahead and pull up this 6N1K in our PDB, in our PyMOL, I mean. So fetch 6N1K. And it's going to open it along with this other one. I could do things where maybe what I want to do is I want to action, and I can actually align this to a molecule. I'm going to align it to this other thing. And so you can see we're looking at just this portion over here. I'm going to go ahead and hide that old structure so we don't have to worry about it. And now let's focus on this one. And I can see up here, the sequence has been replaced by this one for this other, the new structure I pulled in. I'm going to go to color it. I'm going to go to color by chain. Now you can see these chains more easily. I'm also going to change the background black to black so you can see things more easily on the screen. So let's go to display, background, and I'm just going to do black. So now you can see the lettering more easily. And you can see the fact that some of the letters are still grayed out. So even though this is the full chain length, you can see that there are regions where there's actually missing amino acids. And if you come to these regions, you might see like dotted lines indicating that there's a sequence there. There's protein here, but we can't actually see it because the structure was not well enough. The data there wasn't strong enough to model, and so we get these kind of missing regions. And depending on how big the missing region is and what settings you have, you might see those places as dotted lines. And so right now I have it set as chains. And you can see that everything is colored by a different chain. If I wanted to look at a sort of chain individually, say I wanted to make a separate object and control each of these chains individually, I could do things like I could go to over here, instead of residues, I go to select chains. Now, if I click on a chain, I can have a separate molecule. And maybe I want to actually go and I want to save that as a new selection. I could come over here and I could go to action, rename selection, and I want to just go to this chain, which is not a good name, but I can have this chain. And now if I come over here to selection, I can control this chain separately, just using the commands over here. Or I could come over here and I could click on this, and that would be showing up as a selection, and I can control it that way. But if I make a new selection, then that selection will go away. So it's helpful to name your selections so that they'll show up in this command bar and you can deal with them individually. Again, up here, you'll see each of the different strands is going to be given a different like starting, so A or B. You'll see their sequences from N to C-terminus, so the numbers will keep starting over. At the end, you'll get small molecules. You'll get things like those iron molecules. You'll get various other things, various other chains, but typically your protein chains, your main chains are going to be like the first things you'll see. If you want to click to select a whole chain, you want to come over here to chains. Often you just want to come over here and you're going to go to residues. Sometimes you want to do atoms if you're trying to measure distances, but residues is typically your basic bet. Remember to save your scenes, save your sessions. If you want to make an image, come over here to draw array. If you want to change the color, go to C. If you want to label things, go to L. Hide things, H. Show things, S. And do other sort of things like move, align, find distances. You want to go to the A. To zoom in, pinch pad. To go from side to side, translate, option, click and drag. And to rotate, you want to basically use your left mouse click or 2-finger click and then move the top finger around. And if you want to do slab, 2 fingers up and down. That is the basics of PyMOL. So save your session, close your session, and give yourself a pat on the back.